IB Bio, Animal Physiology, Topic 11, Part 7, Kidney and Osmoregulation B, has its focus on the first stages of kidney function, the functioning of the glomerulus and the proximal convoluted tubule. The essential idea is all animals excrete nitrogenous waste products, and some animals also balance water and solute concentration. Here is the outline of all the movies available for Topic 11. Use this outline to find the movie you need for review. This movie is focused here. Here is an image of a kidney. The kidney is an organ of excretion, nitrogen waste, and osmoregulation, water balance. You can see the renal artery bringing blood to the kidney and the renal vein carrying blood out of the kidney. It's important for you to begin thinking about the differences in the blood entering the kidney as compared to the blood leaving the kidney. Certainly, the blood leaving the kidney is deoxygenated relative to the blood in the renal artery. But there's more, a lot more. Pay close attention. Urine, a complex mixture of solutes in the solvent water, is extracted from the blood that enters the kidney, and urine exits the kidney in the ureter. The urine moves to the bladder. In this diagram, you can see the paired kidneys. Blood arrives to the kidney in the renal artery from the aorta, and blood leaves the kidney via the renal vein to the vena cava. And you can see how the urine produced in the kidneys leaves the kidneys in the ureters to the bladder. Here's an X-ray of the abdominal cavity showing the position of the paired kidneys in the body. The image nicely displays the ureters carrying the urine to the bladder. Once again, here are the paired kidneys in the body, the ureters, the bladder, and the urethra. Urine moves from the kidneys to the ureters, to the bladder, and exits the body through the urethra. Here's the first important IB syllabus statement of the movie. Draw and label a diagram of the human kidney, including the cortex, medulla, pelvis, ureter, and renal blood vessels. The kidney has important regions that I'm introducing here for the first time. The renal artery is here, and the renal artery is of smaller diameter than the renal vein, resulting in blood arriving to the kidney under relatively high pressure. The outer edge of the kidney is known as the cortex. The region of the kidney just inside the cortex is known as the medulla, and urine collects in the pelvis and makes its way to the ureter. The kidney removes urea, salts, and water from the blood by adjusting the volume, content, and concentration of the urine. You may wonder how it is that materials from the blood can be extracted and removed from the body. But the basic concept is not that difficult. Blood vessels with small pores lie adjacent to vessels where fluids can leave the body as urine. And right through here, filtration occurs. Small solutes and water pass into the urinary tubules. Large molecules and blood cells cannot pass and continue in the blood. Then the blood vessels and the urinary vessels run side by side to allow a final opportunity to exchange materials. Valuable molecules are returned from the urine to the blood, and waste molecules are secreted from the blood to the urine. A lot of active transport happening here. Again, but with some added vocabulary, here's the fundamental mechanism by which urine is formed. Pay close attention. Blood enters the kidney, branching into numerous arterioles that become a ball of capillaries known as a glomerulus. The glomerulus is surrounded by a capsule known as Bowman's capsule. Bowman's capsule is the beginning of the renal tubule and the formation of urine. Now, ultrafiltration occurs in the glomerulus, where small molecules and water enter the renal tubule. They leave the blood and enter the renal tubule. Then the arteriole exits the glomerulus as the efferent arteriole, and then this runs parallel to the renal tubule. As the blood runs alongside the renal tubule, the blood can reabsorb important molecules like glucose, amino acids, and water. Other wastes, including ions, such as hydrogen ions, are secreted into the urine. The secretion of hydrogen ions into the urine helps regulate blood pH. Any materials that are not reabsorbed are excreted as urine. Now, a couple of things before I leave this slide. Notice how the urine becomes more and more and more concentrated. See the color change. Keep this in mind. Secondly, the functional unit of the kidney is called the nephron. The nephron. Here is a simplified image of the entire nephron. Hopefully, the transition to this slide from the last one is reasonably clear. The afferent arteriole brings blood to the glomerulus. The glomerulus is embedded within Bowman's capsule. Ultrafiltration occurs here as small molecules, including urea and water from the blood, enter the nephron. The fluid moves from Bowman's capsule to the proximal convoluted tubule, to the loop of Henle, to the distal convoluted tubule, and then to the collecting duct. The collecting duct is the last opportunity for the reabsorption of molecules from the urine and then returning them to the blood. Remember that the blood leaves the glomerulus in the efferent arteriole, but it runs in capillaries that are parallel to the tubules of the nephron to provide for reabsorption of important molecules and the secretion of waste molecules. The capillaries that surround the tubules of the nephron are not shown here. For now, work to gain control of the names of the structures. I'll be displaying more complex diagrams just ahead, and I'll be explaining functions of all structures sequentially. Be patient, pay close attention. So let's zoom out to look at the position of a nephron within the larger kidney structure. The nephron can be seen here, and the tubules of the nephron span the cortex, the outer region, and the medulla. This is important. I'll speak more about this later. The urine that exits the nephron through the collecting duct drains into the pelvis and then into the ureter and then on to the bladder. The glomeruli, and there are many of them, lie in the cortex region of the kidney. In this image of the nephron, we can see the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and the collecting duct. Only the loop of Henle and the collecting duct dip into the medulla of the kidney. Fun fact, there are over 1 million nephrons in the kidney. So while this drawing nicely shows the position of a nephron, with the glomerulus right here in the cortex, and the rest of the structure dipping into the medulla, with the collecting duct reaching the pelvis, this nephron is truly a microscopic structure. Once again, this diagram shows the position of the nephron within the larger kidney structure. The glomerulus is here in the cortex. Keep in mind that there are approximately 1 million nephrons within the kidney. Here is the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and the collecting duct. Only the loop of Henle and the collecting duct dip down into the medulla, and you can see how the collecting duct empties urine into the pelvis, leading to the ureter, leading to the bladder. Now these lines we see here, called renal pyramids, represent the thousands and thousands and thousands of loops of Henle and collecting ducts. Here is a micrograph section of a kidney. The cortex and the medulla are labeled. The arrows indicate the general flow of urine toward the pelvis and then on to the ureter. Here is a nice diagram of the kidney to study. You can see the nephron here. The glomerulus is located in the cortex. Here is the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and the collecting duct. Only the loop of Henle and the collecting duct dip down into the medulla of the kidney, and you can see how the collecting duct empties urine into the pelvis, leading to the ureter. Let's pause and look at a diagram that provides some big picture information. I showed you this diagram at the tail end of kidney osmoregulation A. We take in nutrients through the digestive system and oxygen through the respiratory system. Undigested material leaves the digestive tract as feces. Important nutrients and oxygen enter the bloodstream, the circulatory system. Blood releases carbon dioxide to the lungs. Carbon dioxide is eliminated as a metabolic waste of respiration. Carbon dioxide is not the only metabolic waste. We have nitrogen waste known as urea. The blood is filtered by the urinary system, with certain important nutrients returned to the bloodstream. The urinary system excretes water, that's osmoregulation, and the urinary system excretes urea, a metabolic nitrogen waste product. Here's a relevant IB syllabus statement. Annotate diagrams of the nephron, including the glomerulus, Bowman's capsule, proximal convoluted tubule, loop of Henle, the distal convoluted tubule, and the relationship of the nephron to the collecting duct. I've not yet displayed the most complex diagrams of the nephron. I'd like you to use this image to review nephron anatomy in response to the last IB syllabus statement. Before moving to those complex diagrams of the nephron, there are 3 or 4 things I want to mention about this diagram. First, the blood vessels that surround the nephron, capillaries that emerge from the efferent arterioles surrounding the nephron tubules for the purposes of reabsorption and secretion, are not shown here. Secondly, there is a functional relationship between the loop of Henle and the collecting duct. This is an important subject reserved for Part 8, Kidney Osmoregulation C. Third, remember that the glomerulus, the proximal convoluted tubule, and the distal convoluted tubule are located in the cortex of the kidney, while the loop of Henle and the collecting duct dip into the medulla. And lastly, interstitial fluid, fluid that lies between all cells everywhere, surrounds these tubules. In this image of a nephron, there is no collecting duct. Can you annotate a diagram of the nephron, including the glomerulus, Bowman's capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule, and the collecting duct that drops down into the medulla of the kidney? The loop of Henle and the collecting duct have a close functional relationship. More on this later. For now, here are the 4 fundamental functions of the nephron. I'll be providing more detail on these functions in the slides to come and in the next movie. Take a moment to read about these processes: filtration, reabsorption, secretion, and excretion. Here is a diagram that better displays the complexity of the nephron. Blood from the renal artery enters the kidney and branches into numerous arterioles that become glomeruli. 1 million nephrons per kidney. Ultrafiltration of the blood occurs in the glomerulus within Bowman's capsule. The blood leaves the glomerulus to become capillaries that surround the nephron before exiting in the renal vein. The close relationship of the nephron and all of these capillaries allows for the reabsorption and secretion of molecules. Now, the proximal convoluted tubule is here, and this is where the reabsorption of nutrients such as glucose and amino acids occurs. The loop of Henle is seen here, and I will talk more about its function in the next movie. The distal convoluted tubule is here. The secretion of waste molecules or ions occurs in this location. Then this is the collecting duct. The role of the loop of Henle and the collecting duct together will be the subject of the next movie. Here's a chance to practice the annotation of a nephron. With the exception of number 6, which is a small vein known as a venule, you should be able to name all other structures. Turn the movie off now for practice. Here are the named structures. Spend some time with this slide. Turn the movie off again. As a note for the future, fluid surrounds all of these capillaries and tubules. The fluids surrounding all of these tubules is called interstitial fluid. So now we move to some high detail. Here are the next 2 IB syllabus statements. Describe the ultrastructure of the glomerulus and Bowman's capsule for ultrafiltration. Explain the process of ultrafiltration, including blood pressure, fenestrated blood capillaries, and basement membrane. The blood arrives to the glomerulus under high pressure as the afferent arteriole is wider than the efferent arteriole. The capillaries of the glomerulus have pores, fenestrations, or you could say clefts, between the cells of the capillary. The fenestrations, the pores, serve to physically filter the blood. Small molecules and water can pass through those capillaries into Bowman's capsule, but large proteins and red blood cells cannot. The wall of Bowman's capsule is also adapted for filtration. And as well, there is a protein layer, like a protein fabric, called a basement membrane that adds another filtration layer. Once again, small molecules such as glucose and amino acids can pass through the fenestrations of the capillary to become filtrate, while larger proteins and red blood cells cannot. This is a terrific image of the glomerulus. You can see the fenestrations of these glomerular capillaries that are exposed. The blue layer that surrounds the capillaries is the inner wall of Bowman's capsule. You can see how tightly wrapped the capsule wall is around the capillaries, resulting in slits that also serve the role of filtration. The capsule wall is composed of cells called podocytes, in case you're interested. Now the proximal convoluted tubule is here. Notice the microvilli that line the inner membrane. High surface area serves the role of reabsorbing important nutrients from the filtrate and returning those nutrients to the blood that is running in the capillaries surrounding the nephron. Here is another image of the glomerulus surrounded by Bowman's capsule. This is the outer wall of the capsule here leading to the proximal convoluted tubule. You can see the numerous microvilli that line the cells along the proximal convoluted tubule. But the inner wall of Bowman's capsule, composed of podocytes, is tightly wrapped around the capillaries of the glomerulus, creating slits that serve the role of filtration along with the fenestrations in the glomerular capillaries. One more diagram of the glomerulus. The wider afferent arteriole allows substantial blood to enter the glomerulus, maintaining high blood pressure. Fenestrations in the capillaries, the podocyte slits of the capsule wall, and the basement membrane, a protein fabric, all serve the role of ultrafiltration. Here is a micrograph of the cortex of the kidney. Keep this in mind. You can see a few glomeruli here and here and here and here, and surrounding those glomeruli are tubules, proximal or distal convoluted tubules. We can't tell exactly which is which. Remember, the loop of Henle and collecting ducts are seen in the medulla. Here is another micrograph, more magnified, that shows the glomerulus and the afferent arteriole. Another micrograph of the glomerulus, you can see the afferent and efferent arterioles. Ultrafiltration occurs in the glomerulus. I'm still focused on these 2 IB syllabus statements. Describe the ultrastructure of the glomerulus and Bowman's capsule for ultrafiltration. Explain the process of ultrafiltration, including blood pressure, fenestrated blood capillaries, and basement membrane. The ultrafiltration that occurs at the glomerulus within Bowman's capsule is a physical filtration. The fenestrated walls of the capillaries and the basement membrane that would lie here, and the podocyte slits formed by the wall of Bowman's capsule, serve to allow small molecules to pass while preventing large proteins and red blood cells from getting through. If the kidney suffers damage, then urine would have red blood cells and large proteins. To diagnose kidney disease, doctors test urine to see if there are cells or proteins in the urine that shouldn't be there. Here is a nice image showing the tight relationship of the glomerular capillary and the inner membrane of Bowman's capsule. The fenestrated capillary and the podocyte slits of the capsule wall both serve ultrafiltration. In this micrograph of the interior of Bowman's capsule, you can see the filtration slits of the podocytes that form the inner wall of Bowman's capsule. In this diagram, you can see the fenestrations of the glomerular capillary. You can see the basement membrane, a protein fabric that lines the capillary, and the podocyte slits of Bowman's capsule. All three serve ultrafiltration. In this diagram, the capillary with blood is here. Here is the opening in the capillary known as a fenestration. Here is the basement membrane, a protein fabric. And here are the slits formed from the podocyte membrane of Bowman's capsule. The fluid that is physically filtered from the blood is called filtrate and flows into the proximal convoluted tubule. Here is a micrograph of glomerular capillaries along with the wall of Bowman's capsule. Here is the capillary with its fenestrations. Here is the basement membrane just outside the capillary wall. And then here are the filtration slits formed of the podocytes of the inner membrane of Bowman's capsule. Remember that red blood cells and large proteins cannot leave this capillary. Only small molecules can exit to become filtrate in the nephron. Here's another micrograph cross-section. The blood is flowing here. These are glomerular fenestrations. Here is the basement membrane, a protein mesh layer, and then the filtration slits formed of the podocytes of the inner wall of Bowman's capsule. Remember, red blood cells and large proteins cannot leave the bloodstream, but small molecules can exit to become filtrate, entering the nephron. Let's zoom out to look once again at the glomerulus surrounded by Bowman's capsule. The structures involved in ultrafiltration are provided here. I'll let you study this slide on your own. Here is a micrograph of a glomerulus surrounded by Bowman's capsule. The filtrate formed by ultrafiltration flows into the proximal convoluted tubule, and that brings us to the last IB syllabus statement of the movie. Explain the reabsorption of glucose, water, and salts in the proximal convoluted tubule, including the roles of microvilli, osmosis, and active transport. Here's an image of the glomerulus, Bowman's capsule, and the proximal convoluted tubule, and I want you to notice the close relationship of the proximal convoluted tubule and the blood vessel that emerges from the glomerulus. The relationship between these two structures is important as I move to the next slide. In this diagram, the filtrate flows along the proximal convoluted tubule, which is paralleled closely by a capillary. In cross-section, the proximal convoluted tubule can be seen to have microvilli. The flow of small molecules into the blood capillary can also be seen. This is reabsorption. All of the glucose and amino acids are reabsorbed. 80% of the salts and water are reabsorbed from the filtrate back to the blood. This diagram displays the proximal convoluted tubule in cross-section, and you can see the close relationship of the proximal convoluted tubule to capillaries that surround the PCT. You can see the microvilli that increase the surface area of the inner membrane of the proximal convoluted tubule. High surface area, osmosis, and active transport all serve the role of reabsorption of glucose, amino acids, and water back into the bloodstream, so that these substances are not lost from the body in the urine. This is a diagram of a single cell from the wall of the proximal convoluted tubule. The blood capillary is on the left side, with the endothelial cells of the capillary shown here. So the blood is flowing here. A basement membrane lies between the cell of the proximal convoluted tubule and the blood capillary. Now the cell of the proximal convoluted tubule has microvilli to increase the surface area in contact with the filtrate that would flow here. Numerous mitochondria provide ATP for active transport. Osmosis and active transport serve to recover important molecules from the filtrate and move them back to the blood. This is called reabsorption. Here is a diagram of a single cell of the wall of the proximal convoluted tubule. The role of the microvilli, the numerous mitochondria, and the pinocytotic vesicles should be clear to you. The filtrate is flowing here, blood is in a capillary located here, and valuable molecules are being reabsorbed from the filtrate to the blood. Here is a micrograph of the proximal convoluted tubule and the capillary with red blood cells. Individual cells of the tubule wall can be seen each with a nucleus. You can see the microvilli on the filtrate side of the tubule cells. You can also see numerous mitochondria labeled M. Reabsorption occurs as directed by the arrow. The cells of the proximal convoluted tubule are adapted for reabsorption because, 1, they contain lots of mitochondria to produce ATP for active transport. Secondly, they have a large surface area in contact with the filtrate due to the microvilli. And lastly, the proximal convoluted tubule is in close proximity to blood capillaries. Now, in healthy individuals, the urine is free of glucose, large proteins, or red blood cells, but disease exists. If the glomerulus or tube membranes are damaged, then large proteins or red blood cells might be in the filtrate. And diabetics have excess glucose in the blood and thus excess glucose in the filtrate. In other words, not all the glucose in diabetics can be reabsorbed. Here is a table showing the concentrations of various substances in the blood from various regions of the kidney. Here are the concentration units, but let me just refer to the numbers without the units for clarity of communication. In a healthy person, blood arriving to the kidney has 100 units of glucose, and the filtrate has 100 units of glucose, but urine has 0 units of glucose. All the glucose that passes through the glomerular filtration as filtrate is returned to the blood in the proximal convoluted tubule. All of the glucose is returned to the blood except for a small quantity used up during cell respiration, accounting for the small drop in glucose concentration in the venous blood leaving the kidney. Also notice that there are no proteins in the filtrate. Proteins are too large to become part of the filtrate. And last but not least, notice how the filtrate has a urea concentration of 30 units, but that urine has 1,800 units of urea. The urea becomes highly concentrated in the urine as water, the solvent, is returned to the blood. The solute quantity is increased when the solvent quantity is reduced. The venous blood leaving the kidney has significantly less urea as compared to the arterial blood that entered the kidney. The kidney serves to regulate water balance and excrete metabolic wastes. And that brings us to the end of IB Bio, Animal Physiology, Topic 11, Part 7, Kidney Osmoregulation B.